my name is Kaya Govek. I work in Pablo Camara's lab at the University of Pennsylvania, mostly working in single-cell RNA sequencing and then starting to specialize a little bit more in the spatial omics. So I'm very interested in this whole lecture series. So as Dr. Fan mentioned, I'll be presenting our recent project, and it is currently on our archive, yes, titled Single-Cell Transcriptomic Analysis of Multiplexed Immunohistochemistry Images via Antigen Mapping. Can I move? There we go. So because this is a spatial omics group, I know that most people are probably familiar with the technologies imaging mass cytometry and multiplexed immunohistochemistry, which are both technologies that can image a panel of antibodies in a tissue section at single cell resolution, which is especially useful for studies that look at the tissue architecture and cell-to-cell interactions in the microenvironments of cancer or in patients with diabetes. This work particularly looks at immunophenotyping, which involves identifying the cell types present in a tissue section, and then looking at their relative spatial positioning and their expression to determine how those cell types might be interacting in that tissue section. There have been recent advances in this type of technology that allow for much larger protein panels. I believe there will be a talk on this in the next series, which I'm very excited about, but CODEX stands for co-detection by indexing and is a highly multiplexed immunohistochemistry technology that can measure up to around 50 antibodies in a tissue section at sub-micrometer resolution. So that definitely hits the single cell mark. It does this usually using successive cycles of fluorescent imaging where 2 antibodies get imaged per cycle. Once the images are taken by any of these technologies, typically the first step would be identifying which cell types are present in those images. This would historically have been done via something called manual gating, which is done using these biaxial plots where 2 antibodies are plotted on each axis, and then you essentially draw a box around your cell type of interest based on which markers it has high or low expression in. This approach depends pretty highly on which antibodies you've included in your panel, because you are restricted to now around 50, but historically many fewer. And it might not quite be able to identify all of the nuanced cell types present in a tissue section, especially cell states, especially cell types for which you don't have a marker in your panel, or that might be marked by much smaller changes in protein expression. This type of manual annotation is also extremely labor intensive, especially for these much larger protein panels. So recently, more automated gating techniques have been created. For example, XShift, Phenograph, SPADE, or VSNE. These are all visualization or clustering techniques for this type of data, which can automatically separate out cell populations. However, it still requires manual annotation to assign the cell types to those cell populations, which means you might still run into the same problems of identifying nuanced cell types or states that are identified by very small changes in protein expression. On the other hand, single-cell RNA sequencing, which I'd mentioned our lab deals with very closely, allows for much higher resolution in cell states because we can profile the expression level of many genes over the entire transcriptome. There are even some recent single-cell RNA sequencing technologies like CiteSeq, ReapSeq, and AbSeq that can measure gene expression and a panel of antibodies. They do this by attaching a barcoded oligonucleotide to the antibodies, which can then be sequenced along with the mRNA. We'll be focusing mostly on CiteSeq, But all of these technologies are very promising in that they can help us know how changes in gene expression affect changes in protein expression, which can help bridge the gap between these single-cell RNA sequencing technologies and the immunohistochemistry and cytometry technologies. So our goal is to do that. We want to use the data from CiteSeq to help improve and automate the annotation of multiplexed immunohistochemistry and imaging mass cytometry data sets. The foundation of this is that there are many single-cell RNA sequencing atlases for a lot of different types of tissues and organs, which profile the gene expression associated with cell types in those organs. So we could build a similar CiteSeq atlas that also includes the expression of a panel of proteins. And then, since we know that gene expression and protein expression are linked, if two cells have similar antigenic profiles, they likely have similar transcriptomic profiles and cell types and states, which means that we can use cells from CiteSeq to identify similar cells in the multiplexed immunohistochemistry or imaging mass cytometry data sets and map their cell types so that we can annotate the gated populations from those data sets. So we built this computational method. We call it STEVIA for spatial transcriptomics via epitope anchoring. So the first step in STEVIA is to build or procure a CiteSeq atlas for your tissue of interest. In the future, there will be more publicly available CiteSeq data sets, but it's still a relatively recent technology. So we did have to build our own, so I'll walk you through that process. Then we take the protein panels from both CiteSeq and the immunohistochemistry or cytometry technology, and we standardize and merge them into a shared protein space so that we can identify cells that have similar antigenic profiles. And then we use those cells to transfer the cell type information and transcriptomic expression from the CiteSeq atlas onto the immunohistochemistry or cytometry data set. Using that cell type information, we can revise the cell populations from gating, and we can add our own annotations to those. So we decided to demonstrate our method using the Codex data set from the original Codex paper from 2018. That data set used an antibody panel of 30 markers to profile mouse spleens. So we built a matching CiteSeq atlas using the same 30 antibody panel to profile matching mouse spleens. This is a visual representation of that atlas. It's a UMAP representation. So each dot in this image represents a cell from our CiteSeq dataset, and two cells are closer together in the image if they have more similar transcriptomic profiles. So you can see that the cell types separate out pretty nicely in this. So we performed clustering on this data set using an in-house consensus method on a density-based clustering, which identifies 17 general cell populations from the splenic cell types. But we can see that there are some much larger populations, like the B2 cells, which still have a lot of variation in gene expression. And so we used a method from our recent 2019 paper that just got published in PLOS Comp Bio, which uses the Laplacian score to identify differentially expressed genes within a cluster. So when we run this on the B2 cells, we identify genes like CD83, Notch2, LY6D, that can all identify subpopulations of B2 cells based on the maturity or location in the spleen. So if we add those annotations into the existing clusters from our atlas, Here I've shown them using dotted lines, and the italicized text are our new annotations. We now have a total of 30 cell populations in this atlas, which makes it a very powerful single cell RNA sequencing atlas of the mouse spleen, even not including the rest of the context of this presentation. It's a pretty powerful resource on its own. But it's site-seq, which means that we also have this expression of 30 antibodies along with the rest of the atlas, which we now want to use to connect the cell types from this atlas and transfer them onto the original codex data set from this paper. So 10x, which is the sequencing platform we used for site-seq and codex, use very different values to represent protein expression. One is sequencing and one is fluorescent imaging, which makes comparing antigenic profiles between the two data sets actually quite a difficult problem. So the first thing we needed to do is standardize or normalize the expression from both technologies so that we could compare them. And then we used a batch correction method to merge both protein data sets into a shared space so that we could identify cells with similar antigenic profiles between the two technologies. So starting with the normalization, we used a two-component mixture model where one of the components fits to the background peak and the other component fits to the signal peak of expression. And then we just keep the expression from the signal peak. So on the left here, you can see we've plotted a CD4 expression on the codex tissue section. This is the raw expression. And then on the right-hand side is our normalization of the CD4 expression. And you can see that the signal in the T cell zones for CD4 really pops out after our normalization. So we run this on both site-seq and codex, and then we perform batch correction using a strategy that is built on the Seurat anchor correction, which came out in 2018. Using this batch correction strategy, both codex and site-seq get placed into a shared protein space where cells are closer in this space if they have more similar antigenic profiles, now regardless of whether they're from site-seq or codex, which means using this space, we can identify site-seq cells that have similar antigenic profiles to a given codex cell. To confirm that these pairings between site-seq and codex are consistent and to see how in the future the size of the reference data set or the antibody panel we can use might affect this mapping between the two technologies, we ran the same normalization and batch correction using a subset of the data. And we see that as we decrease the size of the antibody panel or the reference data set, the number of codex cells that do get mapped to by any site-seq cell decreases. So the number of annotated cells in codex goes down, but the correlation between their annotations and their original annotations stays pretty high. So we can be confident in the cells that do end up getting annotated, even as we decrease the size of the panel or the reference data set. So now that we know which site-seq cells have similar antigenic profiles to codex cells, we can take the cell types of those site-seq cells or their transcriptomic expression and associate them with the codex cells, which can inform our analysis of the codex tissue sections. So the first thing this allows us to do is actually just to plot which codex cells have a similar antigenic profile to a given site-seq cell. So on the right here, Each panel is a hand-drawn version of one of the codex tissue sections. The T cell zones are outlined using the solid line, and the B cell zones are outlined using the dotted line, and then the rest would be the red pulp. So we can see first that our prediction of where similar cells occur in the tissue section confirms the general splenic compartments that we know. So the T cells show up in the T cell zones, along with most of the dendritic cells. CD4 dendritic cells show up in the bridging channels between the T cell zones and the red pulp, which is known, but is a fairly recent discovery. Monocytes show up in the red pulp, as we would expect, and then the B2 cells localize in the B cell zones. But we see a few different localization patterns within the B2 cell cluster. So we plotted cells with similar antigenic profiles on all three of the tissue sections profiled in the original codex paper. And we see that these patterns are consistent between all of the codex mice. So we can also take the gene expression from these site-seq cells and plot it on the associated codex cells to computationally kind of predict where gene expression will show up in the tissue section. So we've chosen two markers here. IL-1B is a marker for a few red pulp cell types like macrophages and monocytes. BHLHE41 is a marker for B1 cells. So you can see that just from these left-hand predictions, they show up where we would expect. IL-1B shows up in the red pulp, but not at all in the B cell follicles, whereas BHLHE41 shows up in the B cell follicles or mostly around them. We also validated these predictions using single molecule RNA FISH. So on the right-hand side, the upper plots are the actual FISH images. Blue is DAPI, red is CD79, which is a B cell marker, and then the green is our marker of interest. Since the green is a little bit hard to see, we also have hand-drawn renderings of these FISH images, where now the B cell zones are outlined using dotted lines, and then the red cells are the ones expressing our markers of interest. So we can see that it's a little bit clearer in these plots. These FISH images do confirm our predicted expression of these markers. So with this information, our goal would be to identify cells that are interacting in some way in the spleen and identify how they're interacting. The first step there is just to identify what cells tend to be near each other in the tissue section. So again, we use one of our methods from the 2019 paper that we published called the Adjacency Score, which identifies co-localized features in an image. So the heat map on the right shows the significance of this score for every pair of site-seq cell types. The heat map is red if cells from those cell types are nearby each other often in the codex tissue sections. And then there's a black box around it if that significance can't be explained by any low mapping accuracy between those cell types, and I'll explain a little bit about that later. So we can see that this heat map confirms what we know about the general splenic compartments. Again, the red pulp cell types tend to co-localize amongst each other, and the B cell and T cells tend to just co-localize within themselves. as we would expect from our knowledge of the T cell zones, the B cell zones, and the general red pulp. We also see some co-localization that occurs across multiple splenic compartments. So here we can see that the CD4 dendritic cells co-localize with the T cells and the NK cells, which confirms their localization in the bridging channels between the T cell zones and the red pulp, and their role in mediating across different splenic regions. So once again, we did this analysis on all 3 of the tissue sections profiled by codex, and we find that our results are very consistent across all of the codex mice. So we've been running the adjacency score on the cell populations, but we can also run it on genes, especially pairs of ligands and receptors, to see if they are expressed by nearby cells. And so we first identify potential paracrine signaling interactions that might be occurring in our data set using a computational method called cell phone DB, which compares differential gene expression to a database of ligand receptor pairs to identify potential interactions in the data set. The downside of this method is it doesn't take into account whether those ligands and receptors are actually being expressed by nearby cells in the data set, or where those cells are. So we then filtered those interactions to only those ligands and receptors that are co-localized in the tissue section using our adjacency score. So in our preprint, we used that to describe the differences between how red pulp macrophages and monocyte-derived macrophages interact with the different red pulp cell types. Specifically, we identified that red pulp macrophages might be more specialized in positive regulation of the innate immune response in the spleen. So up until now, we've been using the cell types from our mRNA analysis and plotting those and the transcriptomic expression on the tissue, codex tissue slides. But there's no guarantee that the cell types from our mRNA atlas can be equally accurately mapped onto codex using this panel of 30 antibodies. So to assess how accurately a site-seq cell is mapped onto codex, we take the correlation between the protein expression profiles of the codex cells that site-seq cell gets mapped to, which is essentially a measure of how condensed or sparse the region it gets mapped to is. And we see when we plot this accuracy of mapping over the site-seq UMAP, that certain cell types like B2 cells or T cells are much more accurately mapped than other cell types like B1 cells or dendritic cells. So to account for that, we developed a clustering algorithm that takes into account the resolution of cell types that's provided by the site-seq mRNA atlas and how well those cell types can be mapped onto codex. The way we do this is we identify clusters in the site-seq mRNA space using the same method as we did previously. We map those clusters onto the codex cells, and then we compute the Louvain modularity of those clusters in the codex protein space, which essentially measures how consistent those clusters are after having been mapped. This provides us with a joint clustering of both codex and site-seq, where we have shared cell types between the two data sets. Our analysis of our CiteSeq and Codex data sets shows 17 phenotypically different cell types. This is the same number of clusters, but they're actually different cell populations. So we can see that the macrophages and monocytes are a little bit harder for us to resolve using this panel of 30 antibodies. But on the other hand, the T cells and the B2 cells can split into 3 subclusters each, like the regulatory T cells or the marginal zone B cells. So on the left here, I'd shown this briefly on the past slide, but this is a UMAP of the codex protein space. So each dot here is a codex cell, and then they're closer in the image if they have more similar antigenic profiles. So we can see that our CiteSeq mRNA clusters actually map to pretty distinct regions in the codex protein space. We can use these cell types that were guided by the mapping, and we've annotated using our CiteSeq atlas to inform annotations for the gated populations of some of these automated gating approaches like XShift, SPADE, and phenograph. This kind of graph is pretty standard for a SPADE analysis, where each circle represents a gated population, and then there's an edge between populations if they're more similar. We've added pie charts onto this where that represent the proportion of our mapping guided cell types in those gated populations. So for example, this larger green XShift cluster contains most of the B2 cells. So this kind of plot makes it possible to annotate the gated populations based on what cell types those populations represent. There are some populations that represent multiple cell types, and there are some populations that split up the same cell type that could maybe be merged, especially in the SPADE analysis, as you can see, because SPADE forces you to predefine a set number of gated populations. So I'd been talking mostly about using stevia to annotate multiplexed immunohistochemistry or imaging mass cytometry data sets, but it can just as easily be used to annotate non-spatially resolved flow or mass cytometry data sites like FACS or CiteOff. So we've taken the CiteOff data from the original Codex paper, and we've applied stevia using the same CiteSeq atlas to that data set. These are UMAPs of that CiteOff protein space, colored on the right by the XShift clusters, which have then been annotated manually using the markers in the panel. And then on the left is our mapping guided clustering of that data set using the cell types annotated using our CiteSeq atlas. And you can see here that we can resolve just a few more cell types using our automated mapping guided approach with the information from CiteSeq. So just in summary, we developed this method called stevia, which allows us to use data from CiteSeq or ReapSeq or AbSeq to improve and automate the annotation of multiplexed immunohistochemistry or imaging mass cytometry data sets. It also allowed us to computationally stain the tissue section for the expression of genes or the location of cell types. And in doing so, we were able to identify a few candidate paracrine interactions within the tissue section. And lastly, we created our own CiteSeq data set, but our kind of vision for the future is there will be many publicly available CiteSeq or ReapSeq or AbSeq data sets, or even maybe a database of such data sets, which could be used to add annotations to any new incoming multiplexed immunohistochemistry or cytometry data set of a similar tissue type, which would allow for truly automated annotations of those data sets at pretty high cell resolution with no extra need for manual annotation. So with that, I guess a pretty short presentation, but I'd like to thank you for listening to my research. I'd like to thank the Kamara Lab for all their help, especially mIHC, who is the other co-first author on this project. And I'd be happy to take any questions. Thank you.
So thank you, Kaya. This is an excellent talk. And so again, if anyone has question, you can either unmute yourself, I believe you are able to, and ask, or you can use the chat box. Maybe I can ask a question first. It wasn't a question, just I probably didn't really catch, maybe already talk about. So I thought you just used the transcriptome data from the CiteSeq to map to the codex, or use the protein expression map to the codex and then find out the transcriptome data to do cell type annotation.
Yes. So we use the protein information from CiteSeq and codex to identify cells that are similar between the two data sets. And then we map the transcriptomic gene expression from CiteSeq onto those codex cells. So we use the protein expression to connect to the two data sets. And then we use those connections to inform our codex annotations. Does that answer your question?
Yeah, yes. Yeah,
okay.
Yeah, because I think towards the end of that data analysis, you mentioned you compare protein and messenger RNA. So I thought, I thought you were using just the transcriptome data for kind of anchoring the two data sets.
No, there are a few recent, I think Surat came out with one that uses transcriptome information to anchor fish and single cell RNA sequencing. But no, we use the protein information because that's what we have from Codex and CyTOF. They have the protein information.
I think questions in the chat box, you can take a look.
Yes, thank you. Great talk. How do you take into account biological variations across the data sets done by different labs? That is an excellent question. So I guess there's a few different answers or a few different questions there. If it's different labs having done Codex and CiteSeq, then obviously that's the situation we were in. We didn't do the Codex analysis, we only did the CiteSeq. And so it's this, all the way back, it's this normalization strategy that tries to make both protein expression information similar enough between the two data sets. If you're talking about what I was talking about at the end, where we could maybe use existing CiteSeq data sets or a database of CiteSeq data sets to annotate new multiplexed immunohistochemistry or imaging mass cytometry data sets, that's something that is on our list to work on actually, is how to kind of compare information between CiteSeq data sets. So I guess that was a couple different ways to take your question. I guess you can comment or talk if you were intending something different. Let's see, another question. Many molecules have a disparity between gene expression and protein expression. Yes. Does this analysis tackle this in any way, and or does the spatial data provide any resolution to this? So that's a great point to bring up. If I go here. So we did take a look at our CiteSeq atlas. There's a reason in this plot I'm plotting the genes that are typically associated with each protein. So on the top row is the gene, and then the bottom row is the associated protein. And we did do a Spearman correlation between the gene and protein expression. That came out to around 0.3. Which is pretty standard for CiteSeq data. A lot of that is dealing with the sparsity of the data, so it's not quite easy to interpret. So we know that our CiteSeq data is kind of up to par with what CiteSeq data is normally. But at the moment, we don't really have a way for, I guess, fixing, I don't know, like improving the correlation between the gene and protein. Like we don't have a way of checking that necessarily, except for using FISH. I hope that answers your question. Thanks for the great talk. One codex cell may be similar to, yes, to many CiteSeq cells. Since this is a many-to-many mapping, how do you uniquely assign gene expression value for each codex cell? That is an amazing question. Because that's something we spent many days talking about and trying to work on. So right now, the way we do it is each CiteSeq cell is mapped to multiple, or sorry, each codex cell is mapped to multiple CiteSeq cells. And this is weighted mapping. So we essentially just take the weighted average of the CiteSeq expression profiles that are associated to a given codex cell. There are a couple other ways that we thought about doing it, and that's another thing that we're kind of wanting to do in the future, is maybe looking at what, kind of exploring more so what's the best way of calculating the codex expression profiles. Right now, so the reason that we just decided to go with one is we're only using it for plotting. We're not using it for like too many further downstream analysis or statistics or anything. So the different ways of computing the gene expression don't significantly change the visualization. Let's see. Can this framework be extended to disease modeling with spatial details where things are more irregular and unknown? Let's see. So I don't know a lot about disease modeling, so forgive me if I misspeak. I know I was reading a paper the other day that was interested in the use of things like Codex or IMC for modeling like viral cell interactions. And so people are thinking about that. I would say irregular and unknown. So in a lot of our analyses, this is looking at which cells tend to be close to each other in a tissue section. If we don't have enough cells infected, then there's not much we can do to compute the significance of that, if there's not enough for the statistics. But we don't necessarily require just like looking at the visuals to tell what's happening in the tissue section, we did use this adjacency score to look at which cells are nearby each other. I don't know if that answers your question. I don't know a lot about disease modeling, I'm sorry. Cool.
So, Kaya, this is Ron. I have two short questions. So number one, I'm just so curious. I think codex, after you do segmentation, you'll actually get a single cell data, right? That's a pretty accurate single cell data. So I'm just curious if you pull your codex single cell data with your CySeq data, but you only have in codex you only have proteins. In your CySeq data, if you only take proteins, and pull the two data sets together and say, okay, I don't know which is which, we just put them together as one data set and analyze what are gonna see. I'm just super curious. That could be very interesting.
Well, so actually that's pretty much what this is doing. You can
that's
see
right here.
if you're comfortable with the idea of UMAP, so this right hand blue and red is an actual UMAP of CySeq is in blue and Codex is in red. This is a subset of the Codex data set because it's much bigger than CySeq.
Oh, okay.
So that's essentially the point of all of this, is finding a way to combine these two types of data into one space so that we can analyze them together.
But what I thought is, in this case, you know which is which.
That is true.
And then your computational so try to make some effort to merge them together.
Yes.
But what I'm suggesting is you just put the two sets of
just
data
concatenating
together.
them. Yeah. So we didn't try that specifically, but because what I was talking about, there are very different ways of representing the protein. It's not like if I just concatenated the two data sets and I showed you one of the cells, even without me telling you which one was CySeq or Codex, you would be able to know which one is CySeq or Codex. We use these batch correction strategies to do our best to merge the two types of data, because otherwise we can't compare between them. Okay.
That actually leads to another question I have. When you look at the Codex data, right, that's basically just a section of the tissue, not the entire tissue. But how we do single cell RNA sequencing, we basically grind the whole tissue and dissociate into single cells. Basically, we have the cells from all different layers of the tissue sections. If you cut the entire spleen into a bunch of tissue sections, but your spatial data comes from only one of those many sections. So I think in this case, the difference you see here, for example, between the red and blue, probably it's real. That tells you, okay, which cells you are missing in that tissue section.
Yeah, that's a great point. Actually, that's something we're seeing. So the differences between the red and the blue, we know, are both biological cells that we're missing and technical, just because there's still a lot of noise in these data sets. But if I go, so this is the Codex UMAP on the left-hand side. And any colored cells are ones that we have CySeq associations for. But you notice here, I think you can see my mouse, this gray region, there's an entire cell type here that Codex can image because it images all cells in the tissue section, but CySeq can't because we did a manual dissociation of the cells. So we're missing a lot of the stroma cells that we would have picked up via Codex. So that's a really good point. And maybe it's a reason that we shouldn't have used manual dissociation, or that future CySeq studies should try to match the codex data set, but you're always going to have these differences. And the cool thing about this algorithm is we notice that. So it's not like we force those cells to be mapped to CiteSeq and then mislabel them as the wrong cell type. Like we did pick out that there are entire regions of the codex data set that we can't map just because we're missing those cells. Yeah, that's a good point.
Yeah, I agree. I mean, sometimes you can't perfect match the two different data sets, and that's actually a good thing. That means you can find something biological interesting between the two different data sets.
Yeah.
I think I have one more question probably in the chat box.
Yeah. To Rong's point, maybe you could divide the coded data in half. Yeah. So I didn't mention this. That is a great point because we did that. We split it into 8 chunks and mapped each of the 8 individually and then concatenated them, because you're right, it does map a little bit better with 2 data sets that are a similar size. So we did split the codex data set into 8 different pieces that are around the same size as our CiteSeq data set and mapped each of those individually and concatenated the resulting space. Yeah, that's exactly what we did. Yeah. Any more questions? Yeah. To your point, Rong, you had mentioned that sometimes there's biological differences represented by missing cell types. And so this is all wild type information. I think one of the CiteTop data sets we run on is a mutant mouse. But that is a really cool application of this, is identifying maybe cell types that mutant or control might be missing.
Okay.
Yeah. Any other questions?
Okay, if no more questions, let's thank Kaya again. This is a really super interesting talk and bringing, I think, experimental single cell, spatial, and also computational data analysis. This is an excellent talk. Thank you, Kaya.
Thank you.
All right, so this presentation officially ends actually the entire series 1 of our Spatial Omics seminar series, and hope you guys can dial back in starting next week to kick off our series 2. So Ahmet, you have anything to further kind of announce or comment?
I think things will be similar to the first one, but the excitement will continue to grow. And thanks for your efforts putting together the first one and starting this entire community. And with that, I think we look forward to seeing you guys next week.
Yeah, finally, I think all the recordings are uploaded to, I think, Ahmet's group YouTube channel. I also create a YouTube channel, and I will kind of share that link. Also, I will prepare a flyer, and you guys can circulate to whoever might be interested in this spatial omics topic. Thank you very much.
Thank you. Bye-bye.
Thank you. Take care and stay safe.